let's go to the next section. So this is the beautiful virion, and let's turn on the subtitles, shall we? There we go. So, these are two of the three forms of virus. The other one being the intracellular enveloped virus that actually has one more membrane beyond this. So the bare minimum number of membranes that you get on one of these viruses is 2. There's going to be an inner membrane, and you just have to imagine that arrow is stretched all the way down to here. Both these little blue things are membranes. That's just how they draw them in this site. The outer membrane is here, and the outer membrane has some proteins in it, at least some of which are going to be fusion proteins. So it's going to need fusion proteins for its inner membrane, fusion proteins for its second membrane, and then the external envelope virus right here actually has a series of proteins that only appear on that. And so some of those also have to be fusion proteins. So it's like it's got 3 layers of different spike proteins or different GP120 and GP41 proteins, or rather proteins that are doing that function. And together, each of these layers is going to bind something and fuse, bind something and fuse, bind something and fuse, and then finally we're going to dump out our core into the cytoplasm. It's, yeah, kind of a ridiculous way to make a virus, but you could say that about so many aspects of so many viruses, really, now couldn't you? So, yeah. Viral double-stranded DNA surrounded by 2 lipid membranes versus viral double-stranded DNA surrounded by 3 lipid membranes in the external or extracellular enveloped virus, the EEV. Okay, and so these are what the late genes are going to do. They're going to make the virion, so that's what the top up there means, late making the new virus. So the first thing that's going to be done is that we're going to make a protein that's called D13. And D13 is fascinating because D13 is one of these proteins that is what we call a beta barrel protein, or rather a beta jelly roll protein. It is going to make an icosahedral capsid, just like on poliovirus. Just like those VP2 and 1 and 2 and 3 are going to together make an icosahedral virus, and they're all beta jelly roll proteins, and so is D13. And so D13 is going to assemble, is going to, there we go, assemble together. And we're going to pack all the DNA inside the virion first before we assemble it. We'll assemble around it. And so there we are, almost finished, but not quite. And there is the completed version. And then D13 is going to bind to the Golgi. And D13 really sticks to lipids. And so it's already loaded some proteins. Here you can see some little bumps in the Golgi. These are real electron microscope images, by the way. It's a really neat technique where they kind of evaporate some parts of the cell as they're taking the image. It's, yeah, just the coolest looking pictures, I think. And so here it is next to the mitochondrion. It's going to travel over to the Golgi. And there in part E, it's going to wrap around the Golgi and look at that virus. It's the D13 is binding to the membrane. And it's just pulling it, yeah, nice and tight to the surface. And once it's all the way wrapped up, then, boom, it's going to pop. And we're going to undergo hemifusion, then fusion. We're not sure exactly how that happens, whether it's just physics that's doing it at that point, or whether it is some viral proteins. But Poxvirus has so many viral proteins that it could well be. I think the other thing to look at is look how loose the core is here. See all that empty space, the black stuff in there? And as we go on and tighten this thing down, we are actually going to, look how condensed that core is. And the core is going to keep condensing. We're going to keep squeezing out water and squeezing until it is actually misshapen. So it starts out as a perfect icosahedron, a perfect little 20-sided thing. And then it's going to end up totally smooshed and dehydrated. So this dark green right here is actually the D13 protein. And these are some of the other proteins that are going to bind to the outside of that helix. And that's what binds the membrane. And that's what happens to it. It turns into this sort of figure 8 kind of shape. Here you can see some immature virions. So these are the little sort of D13 icosahedral capsids, and they are all trying to be exactly the same size and shape. And then this is after the wrapping down here in E, where you're actually going to get those intracellular mature virions, the IMVs. And these, if you were to break open that cell, could actually come out and infect. And there you can see that the capsid right here, this great big thing, internal, D13, has been flattened. And it's now this sort of orange little band right on there that is the same as what's going on the outside here. So yeah, that's kind of cool. And then there are a bunch of viral proteins that get packed around the center of this little virion. And so yeah, you end up with this kind of brick-shaped or loosely hexagon-shaped virion every single time. And there you can see a whole bunch of them inside the cell. So it's a neat little process. The other important thing about Poxvirus assembly is that assembly is going to occur on the cytoskeleton. And I mean actually on the actin chains of the cytoskeleton. Actin and actin and I don't know, I forget what the other one is. I don't know, my tubulin, there we go. Oh my gosh, yeah. Brain's not working today. Actin and tubulin are going to be the main components of the cytoskeleton. There we go. And so the way actin works is that you will have actin, and it'll have ATP at the end that's going to grow. So this is going to be the old end. We call this the minus end with the ADPs. And then down here, the last two subunits will still have their ATP, and we're going to lose that ATP over time. Actually, I think that should probably be an ADP, and I think it's only the end one that has an ATP, maybe. And so, yeah, it's got, this is the plus end. And so we're going to grow by adding on more little segments to this end. And what the virus is going to do is it is going to sit at one end of the actin and force the cell to start making that actin chain grow. So the virus is going to sit on top right here at the minus end, at the end that would normally break down. So the cell would normally be coming in here and disassembling from this end, but the virus will stick to that so that it can't disassemble. And now as this actin rod grows, it's going to keep growing, the virus is going to be picked up and pushed basically up through the cell. And yeah, we call this thing a comet tail, basically. So imagine the virus is the comet and the actin is a little tail on the end of it. And the virions will, yeah, they'll bind to the ADP end and cause the cell to start to extend this chain. And so the virus can use this as a way to rocket itself from inside of one cell right through the membrane and into the next cell that's next to it. And so this is how the virus can actually spread, by using your own cytoskeleton against you. Yeah, poxviruses are so elegant in the way that they go about all these things. They're just masters of manipulating the inside of a cell. Really neat viruses. Not as cool as coronaviruses, obviously, but, you know, I'm biased. I'm pretty biased. And these Comet Tails are going to move fast. About 3 microns in a minute is ridiculous speed for intracellular processes. And they are known to spread the virus, yeah. And the whole cycle takes about 12 hours from entry until you are basically spearing on the end of your little comet tail the next virus. All right, so we covered a little bit, or we will cover rather, a little bit of the poxvirus host cell interactions shortly. But first of all, this is going to introduce some of this to you. So poxviruses, in addition to binding that actin, and in addition to binding the Golgi and doing all these other cool things inside the cell, are going to be very good at manipulating the intracellular and actually full immune system immune responses. So they are very good at, let's see, they have a protein, or rather Vaccinia virus has a protein called K3L that's going to inhibit the action of a protein called protein kinase R. Protein kinase R is trying to detect, it's another one of these mechanisms that tries to detect virus RNA, like too much RNA or RNA that looks a little bit wrong, something like that we think is what sets it off. And K3L will just bind this thing and shut it down. In addition, there's going to be the E3L, so on segment E, third gene, and it's facing to the left. And that is going to bind double-stranded RNA and sort of cover it up so that PKR can't see it, protein kinase R. Protein kinase R stands for protein kinase RNA dependent, PKR. And this is going to be one of these intracellular immunity triggers. And so, yeah, K3L and E3L shut those down. And there are quite a host of other poxvirus-encoded proteins that will interfere with the actions of the complement cascade. So this is a way that your immune system can shut down viruses and, in particular, bacteria without having to know where they are. As long as you can attach an antibody to them, you can blow them up. And interleukin-1, this is going to turn on the full immune response to hunt and kill the virus. And the poxvirus is able to turn this off by grabbing the IL-1 so it doesn't work, and by making IL-10, which sort of contradicts the order of IL-1 and turns off the inflammatory response. And the virus is also going to bind TNFs, which are tumor necrosis factors. And TNF-alpha is going to be the big one there. That's another one of these very pro-inflammatory cytokines. It sets off a big immune cascade, calls in a lot of different kinds of cells and activates them, and is very harsh but very helpful in clearing out virus infections. And Poxvirus doesn't like it, so Poxvirus shuts it down, like all these other things. And so here are some of the examples. I'm going to go over this in a little more detail in one of the other slides. But everything that's red on this one is a protein from one of these pox viruses. And so they see like E3L, K3L bind protein kinase R and double-stranded RNA. Great. Each one of these grayish white dots is a cellular protein. And you can see most of these are involved in the immune system. So here are parts of the complement system. Here are a bunch of cytokines up here and chemokines, which are both little proteins that are used for communication. Here are a bunch of interleukins, and these are also used for communication among white blood cells. Here we have receptors for some of these signaling molecules, and we have things that will bind to growth receptors or turn off some components of the immune system. Very interesting little proteins. And every poxvirus will actually have its own set of these things. It might have 15 or 20 different ways to mess with or turn off aspects of the immune system that are troubling or a little bit harmful to the virus. So it's very good for the virus, and the virus actually gets these genes by stealing them from its hosts. It will actually take the gene from the host cell, take a broken version of it that doesn't work very well, and yeah, then it will just use those against us. So for example, interferon alpha and beta, these are the general cell's way of signaling distress. So this is the goal of that RIG-I MDA5 pathway. The point is to make interferon alpha. This is the kind that your average standard cell in the body can make. And so they'll do this when they notice there is any sort of virus in the area or something like that. It's a danger signal that calls for help and tells other cells to basically lock their doors because there's a virus on the loose. But when the Vaccinia virus infects a cell, so this yellow one here in the center is infected by Vaccinia virus, what it does is it releases a whole bunch of soluble interferon receptors. So normally interferon receptors are hooked up to that whole cascade. Do you remember that thing? You've got your interferon receptor alpha and beta, you've got your JAKs, you've got your STAT1 and STAT2, and then those are eventually going to go to the nucleus and along with the IRF proteins, and then you're going to finally get a response. Well, what the virus does is it just releases soluble versions of the receptor, and so they bind to the interferon, but they're not connected to anything. They're just floating around. And so it just launches all these little decoys off the cell. And these are the virus's own gene. It actually encodes this. This is part of its genome. And yeah, it shuts down an interferon response by just snaffling up all of the interferon that's out there. Okay, so this is a nice little diagram showing you some of the ways that one of these viruses will block the interferon pathway. So this is just Vaccinia virus, and look at this. Look at this. compare this to coronaviruses, and you can see coronaviruses are, I hate to say this, but they're amateurs. So for example, interleukin 18 could come in, and this could help signal to the cell, but the virus is going to make C12 protein, which is this little red thing. It's a soluble receptor, a decoy for interleukin 18, so bam, it's never going to bind. If we did bind something to TLR2 or TLR6, these are going to be proteins on the surface of immune cells that recognize viral components or general signals of danger. That's fine. We've got the A46 protein that hits MyD88, which is underneath TLR2 and 6, and several other TLRs, and shuts that down. We've got the A52 protein that shuts down IRAC2, and so on. Yeah, E3 is going to block double-stranded RNA recognition by RIG-I. Fantastic. And that may be the 2'O methyltransferase. I can't remember. But anyway, and so on. Let's see. Say you want to activate NF-kappa B. You can't, because the entire process to activate it is blocked by 3 of these other ones, B14, K1, and M2. And you can't even go this other route to activate it. Rather, C6, K7, and N1 are going to come in and block that. And so behind each of these little code numbers, there is an entire story. There's a structure of a protein. There is when is this protein expressed and where in the cell. There is exactly what does this protein bind and how does it do it. But each one of these is, it's like a little monkey wrench that the virus throws into cellular immune machinery and absolutely stops from making interferon alpha or beta or making any of those other inflammatory cytokines, the other interleukins like IL-1 or IL-6. And then even if we do manage to make a little bit of interferon beta or gamma or alpha, they have receptors that will bind to the interferon gamma or bind to either alpha or beta interferon. And so that's the B8 and B18 proteins. And then we've got an H1 protein that's going to come in here and inhibit STAT phosphorylation. So fantastic. If you block STAT1 phosphorylation, you block both of these processes. So yeah, any way that the cell can respond. And so on. You've got various other genes, and you'll notice that some genes have multiple effects. So K1 is going to block I-kappa B alpha chain degradation, and K1 is going to block an unknown interferon stimulated gene, which is going to come down here and act on messenger RNAs and set the cell off, basically tell the cell that there's danger around. So DNA viruses, big DNA viruses like Poxviruses, just contain this absolute arsenal of ways to shut down a cell, and it's really no surprise that once these things get in, they're very hard to get out of your body. All right, so let's go to vaccination now. So vaccination starts out, and before this vaccination, there's a thing called variolation. Remember that the smallpox virus is actually called variola virus. And so the idea behind variolation is that you would take someone who has the variola virus minor strain, the less deadly strain, deliberately pick some of their pustules, like this poor guy has. I think he actually has monkeypox, and inject that into somebody else, or rather put it into their skin. And by doing this, you would definitely give them smallpox, but if they got the mild form of smallpox, they would get over it eventually, and once they were over it, they would be immune for life. Problem is, this is long before any kind of quality control. All you could do was hope that, if this person survived and didn't seem to be in too bad a shape, then you could hope that you had the variola minor strain. But it would be hard to know. And so Lady Mary Wortley Montagu introduced this practice into England from Turkey, as where they started doing it in 1721. There were problems actually identifying whether you had variola virus minor or major, given that this is long before DNA was something we knew about. And so deaths were not uncommon. About 1% of the people who were variolated ended up dying. Yeah. And so Edward Jenner, the founder of vaccination, was actually variolated while at school. Okay. And so that's the climate that into which we're going to have the first vaccination. So we've got this Edward Jenner, he's the big guy up here. We've got James Phipps, who's the little kid right there, and this is from a statue, I think. In the I think that might be in the Royal College of Surgeons, if that's the one I'm thinking of. There we go. May 1796, you got a dairymaid, Sarah Nelms, and who's gonna say, hey, you know, Mr. Jenner, Mr. Virologist, look at this rash on my hand, something like that. Jenner realizes immediately that this is cowpox, and he's heard all these rumors because, you know, before there's the internet, the rumor mill is running rampant, and the rumor is that people that work with cows will often get cowpox, but don't ever seem to get smallpox. And this is before, you know, a lot of statistical tests would have been invented or used in science. And so he was kind of acting on a hunch. But he said, okay, well, let's test this out. So he knew that people get cowpox all the time, and it causes a couple little blisters, but no big deal. And so, the 8-year-old son of his gardener is now old enough that he should be variolated. They should give him the mild form of smallpox virus in order to protect him from dying. Yeah, this is why life was terrible. One of the many reasons why life was terrible back in the 1700s. So he injects him with the cowpox, lets the cowpox take hold, and then, a month and a half later, variolates the boy. So actually injects him with live smallpox virus, and the boy does not go on to develop mild smallpox at all. He actually develops no smallpox. And so from this experiment with an N of 1, that's one person right there who got the vaccine, he publishes his research into smallpox in the form of a book, because why not? 2 years after actually doing it, which I guess that's not so unusual for science. And there we go. It's his little observation that inoculating with cowpox in one person seems to protect against smallpox. And why not? And so he called it the Variola Vaccinia, the cowpox virus, basically. And so Vaccinia, that's where we get the word vaccine, because cow, vaca, and so vaccine, or whatever, however you would say that in Latin. Vaccine, there we go, vaccinae. That is going to be the root word for vaccines, so it's going to come from cowpox virus, just like that. And that's the start of vaccination. And so then vaccination spreads around the world. The World Health Organization gets together. This is one of their big pushes, and in 1965, they actually achieve eradication. But it's only possible to eradicate a disease if it meets these criteria. So it's worth thinking about this when we're thinking about other viruses, when we're thinking about SARS coronaviruses, or polio virus, or the common cold, something like that, or flu virus. So there's no other reservoir for variola virus except for people, including primates. And that was true. And that's pretty rare. If you consider most other viruses, like influenza viruses, grow in all kinds of birds. Coronaviruses grow in other animals. But poliovirus only grows in people as well, so it meets that criterion. Second criterion. Okay, variola virus can only cause acute infections, which means it's going to infect you, you're going to know you're infected, and from that point, you are either going to die or you are going to recover and have lifelong immunity. So it's got to be really immunogenic, and it's got to either kill you or make you stronger, quite literally. And if you meet that criterion, then we can get rid of it. And so there are many viruses for which we do not meet that criterion. And but for variola virus, rather smallpox, we do. And for poliovirus, we're pretty close to meeting that. So polio still seems possible. And lastly, you have to have some kind of effective immunogen, something you can give as a vaccine that will actually work. And in this case, we have Vaccinia Virus, which you'll remember is not cowpox virus. It's the virus that people thought was cowpox Um, but turns out to be a virus from an unknown source, which is still a pox virus and still protects us against smallpox. And so it's still used to this day, even though it is definitely not and has never been cowpox virus. So yeah, I don't know. And so because smallpox met these criteria and because polio meets these criteria, it's possible to eradicate them. Many other things like influenza fail on, let's see, the first criterion. No, birds have every kind of influenza. Second criterion, influenza causes mostly acute infections, but people sometimes die, but the ones who recover can get reinfected again next year. So there's no lifelong immunity. So bam, we failed number 2. And number 3, yeah, there are effective immunogens, the various vaccines that we use. But you can't meet 1 out of the 3. You got to meet all 3 or else you don't get to eradicate a disease. So how do we vaccinate? Well, this is what the vaccination needle looks like. And if you were to go on YouTube right now and Google Vaccinia Virus inoculation, you would see this thing actually being used. So on the tip of it, if I remember right, I think it has 17 little miniature needles. And so you're going to get some virus onto those needles. And then the way that you vaccinate is called scarification. You actually just mash these 17 little miniature needles around, just mash, 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 mash into the arm, because we need to get that virus down to the basal lamina, to that bottom layer, just like with papillomavirus, because that's where it's actually going to grow. And Vaccinia virus and some of these other pox viruses, yeah, it worked like that. So it's then going to grow in those cells, and you're going to start out with a 4 days after, you'll have these little pustules forming. They'll be filling up with little pussy blobs. There's lots of live virus in these little pus blobs. And so it's very important, if you do get vaccinated, to make sure to keep this covered and do not touch it and then touch your eyes. That can be deadly. Because if you do, you can end up with Ocular Vaccinia. So this is Vaccinia from an inoculation in the arm, but where somebody's touched their eye before it had completely healed. And once the scab has fallen off, then generally it's considered to be safe to once again touch your arm without anything else. They used to vaccinate babies against smallpox, which in times of rampant smallpox, that was pretty good. But something like 1 out of every 1,000,000 infants actually gets unchecked spread of the virus after they vaccinate. So here you can see the place where this baby was vaccinated right there on the arm. And you can see that the virus has spread and it's erupting on the skin all throughout the body. Poor baby. And so, yeah, this generalized vaccinia can be deadly. And you wouldn't want your vaccine to actually cause harm, even as it's protecting. I mean, yeah, it's a trade-off. It's a very difficult trade-off. But this is something that people have to look at when making vaccines. Because you need to do something that is strong enough to get the body's immune system's attention, but also mild enough that a person is guaranteed to be able to fight off the virus, even if they have different genetics than some of the other people you originally tested it in. And so it's tricky. And so that's why we don't vaccinate babies with smallpox vaccine anymore, because it takes a while to develop some parts of the immune system, and occasionally, 1 in a million, you'll get a baby that just doesn't and has a terrible disease because of the vaccine. And that's not what any parent wants. So, go back one. Here is smallpox eradication. So 65, we're almost eradicated. We're eradicated in the US and most of Western Europe, but there's still a couple places where there's a little bit of smallpox left in the world. And so you can see 69, 71, narrowing it down, 72, India, Ethiopia, and 74. Bam, just those two. Is it a little Pakistan? No Pakistan. Yeah, good. And then you finally get to 75, and it's only in Ethiopia and Somalia. And this is the guy. This is the last guy called Ali Mal Malin from Somalia. He's the last person to have smallpox, and to catch it in the wild anyway. He caught it, And I believe he caught it on the 26th of October, and he subsequently recovered. He caught the mild strain, variola virus minor, and was fine. And after he had recovered completely, there were no more cases. And so after 2 years of no more cases, finally in 1979, the World Health Organization was able to certify that smallpox was now eradicated from the face of the earth. Now, there were still small stocks in a couple of countries. So the Soviet Union and the US and the UK and a couple other countries in Europe at the very least had stocks of this that they'd stored away and were actually doing some research on it. And around this time, people were told that you're going to have to pack up your research because we've gotten rid of this in the wild. And we need to be stopping. We don't want to have to vaccinate everyone forever because the vaccination is a little bit dangerous. And so we want to just have no more virus in the world. And at least one person got infected as there was a scramble to try and do the last experiments. It was a lady at a research facility in the UK. And she actually died from that. She wasn't actually working on it herself. She was working in a lab downstairs in a dark room, and somebody was flushing their last virus down the sink, and it was a leaky sink. A little bit of it dripped through the ceiling and infected her. Yeah, so that was terrible. All right. But anyway, so now in the modern day, we don't use cowpox. We use Vaccinia virus. And the origins of Vaccinia virus are obscure, to say the least. So let's see, Vaccinia virus is up here, VACV, VACV. And so you can see it's up there. And the other things that are similar branches are going to be, for example, rabbit pox virus is right up here. So there was a time when people thought, well, it might be related to a rabbit pox virus. It's definitely down here in this cluster. And you got a lot of things down here in the cluster. So if we blow this cluster up, that's what you can see down here. It's actually quite far from variola virus. This is where all the smallpox viruses are. Here's ectromelia, the mousepox virus. This is going to be big in the next section of this. Here's cowpox virus, one strain. Here's a different cowpox virus strain. And this is probably the one that would have been used and given protection against variola virus. Here are camelpox virus, and I think these are monkeypox viruses over here, which can infect humans and are one of these emerging agents that people like the WHO are always watching because they could very easily become the next smallpox if they get out of hand. So yeah, they're a thing to be monitored very closely. And so, vaccine strains used to be propagated from arm to arm. So you would vaccinate somebody, and then you would wait for that blister to form at the vaccine site, and you'd take a little pus from that and vaccinate it into somebody else. And they'd also pass this on animals. There are these stories of ships where they needed to take the smallpox vaccine to the New World, to the Americas. And at least according to what I've read, some of these ships, they filled up with orphans. And because the journey was a long time and you couldn't, you had a limited time in which this virus would be infectious, they just used them as a little chain of orphans. They'd infect one, wait for the pustules, use that to infect the next orphan. And so, they used the orphans as a disposable scientific supply, which is, I would say, ethically questionable in the modern day. But it did manage to get the vaccine over to the Americas at a time when it could do some good. So, I don't know. Maybe it's not all bad. Anyway, the DNA sequences show that Vaccinia is closest to, there's a couple other branches up here that aren't shown on this, but buffalo pox, rabbit pox, and there are a couple kinds of horse pox virus. But the actual Vaccinia, whatever its original host was, this virus is extinct in the wild. And so we will probably never know exactly where it came from. Some people are suggesting that it might have been a kind of horse that's currently extinct that was its original host. But they got pieces of it, and it's not quite the same. Close, but not quite the same. All right. That's the end of that section. Thank you very much.